Thanks. Thanks for inviting me. You didn't have much choice, but to follow with the tradition of saying a little bit about my personal background, unlike the last two speakers, I didn't grow up in Essex, but I did go out with a girl from Ilford, not for very long, but anyway. I used to have outside interests, but then when I moved to the MPI in Nijmegen, the other directors won't let me have any outside research interests anymore. But I thought, that's not really true, but I thought I would give you a bit of insights into some major influential figures on my life. I can't show all of them here because there's not enough space, but here are a few of them. And there'll be a glass of wine for anyone who can identify all of the different influences that are shown here. But I just want to point out here that this is probably, you shouldn't really underestimate that what you can learn, the wisdom of an imaginary cartoon tiger. So I'm just going to move on to another figure who's been very influential, and I think most biologists would cite this man as a major influence or a hero of theirs. Darwin, this is Darwin, by the way. Darwin had a lot of interesting things to say about a lot of different aspects of biology and of human biology as well. And actually, this is a nice quote that has been made, or kind of been made quite well known by Steve Pinker's book, The Language Instinct, which I think is actually kind of defangs this a bit, because this is the actual quote in its real essence is quite interesting. So Darwin said, "Language is an art like brewing or baking. It's not a true instinct, for every language has to be learnt. It differs widely from all ordinary arts, for man has an instinctive tendency to speak, as we see in the babble of our young children, while no child has an instinctive tendency to brew, bake, or write." So this is about an instinct to acquire an art, and that's a beautiful way of sort of talking about the interplay of genes and environment in building a trait, as opposed to something that's just genetically determined. To put it another way, we know that we have this kind of predisposition for acquiring complex spoken language. And I'm not going to go over all the evidence that we have for that. But suffice it to say, we know that this is not the same as saying that the language, anything particular about a language, could be embedded in the genes, in the genetic information. Because we know this because a child who grows up in the Netherlands will grow up speaking Dutch if they're surrounded by Dutch speakers, and the same child, if he was growing up in Japan, would learn Japanese. But there is something at the heart of the genome, something in our genetic information that helps us to soak up this kind of environmental input from around us, from our social environments, and places great importance on it. And this is the kind of stuff that could be encoded at the genetic level, and we should be interested in trying to find those effects. Here's Darwin again, and this is the kind of this is kind of his early ideas about the tree of life and about how everything might relate to each other in a kind of huge tree. And one of the problems with studying something like language is I will hear a lot I think later in this course about what we can learn from other animals, and I'm not really going to talk about that today. What I want to talk about really is, if we're limited to just studying this one guy who has the trait in the fully fledged form that we can study, how can we actually make progress without having to rely on looking at homologies with other species? So is there something that we can do? How far can we get just by studying the human animal? And this relies on a general principle that many biologists and geneticists just take for granted, but maybe is not so palatable for people who've been studying sort of generative linguistics or something like that, which is that there is we use variation. We're interested in variation, and we can use variation not in a trivial way of just documenting, oh, look, these things vary or they don't, but we can use it as a way of getting insights into the underlying biology. And the idea here is that we know that there's lots of variability in phenotypes, in the way that people look, the way that people behave, their physiology, their anatomy, their neurobiology, and so on. Now, if we can study also all the variability at the genetic level, we can look for relationships between these two levels, look for correlations, and use these correlations as ways of getting into biology. So it's a way of, it's kind of like, you can see it like hypothesis generating tools, looking for these correlations, because you're not directly showing causation, but it gives you a way of getting into the biology. So what I want to do now is, I'm not going to spend a lot of time in this talk talking about the variability at the phenotypic level. What I want to try and do is, because I think that's something that the psychologists and the linguists and the neuroscientists can probably illuminate better than I can. What I really want to get to is what kind of variability we are beginning to see in genomes, and how we characterize it, and how it can help you guys to push forward these kind of studies of how genes might impact on human language. Okay, so this is not a picture of my collection of multicolored socks. It's actually it's actually a human genome. So we have each of these is a chromosome, chromosome 1, 2, 3, 4, 5, 6, all the way up to 22. And then this is a male, so he has an X and he has a Y, and females would have two X's. And each of these chromosomes comes from one parent. So this chromosome might be from your father, and this from your mother. There are things that can happen to these chromosomes. So there's variations that can happen at a very gross level. So here's examples of a couple of them. And you'd be surprised how many of these kind of strange things are kicking around in the population, because sometimes we don't even know that we're carrying them. And we might not find out unless we had one of these kind of tests where you see all these karyotypes of the different chromosomes laid out. So this type that I'm showing at the top is called a translocation. So these are two different chromosomes. So this might be chromosome 1 and this might be chromosome 2. And a section of one chromosome is broken off and exchanged with a section of another chromosome. And what you might have here is the entire genetic material of the person might be fully intact. So they might have all the genes and there may be no problems. But what would happen if there's at this precise point where the chromosome's been broken, if there was a gene and it disrupted that gene, it split it in two, then that might actually knock out the gene in some way and give a disorder. But as I say, people might sometimes carry these things around and not even know that they've got them. Another and sometimes this is a kind of balance change where no material is lost, but sometimes you could have a rearrangement where some material is also lost when this happens. And so these things here are called deletions. And this is where a whole section of chromosome might be lost. And some of you might have heard of Williams syndrome. That's a classic example. And often the reason that these happen is because there are repeated sections of the genome that kind of get mixed up, and the bits in between get lost. And if there's a bunch of genes in there, you could have a disorder as well. So translocations and deletions. And then you can have, of course, in most cases where there's a deletion syndrome, there's also a syndrome where there's a duplication. So, and you can see here that there's a section here that's become tandemly duplicated. And sometimes things kind of turn around as well. So you have these kind of things like inversions as well. So this is what can go, these are kind of gross chromosomal changes that have been known for some years because people could see them under microscopes. And there are also bigger things like you can have an extra copy of a chromosome like Down syndrome, chromosome 21, or loss of a chromosome, as we see with some sex chromosome disorders as well. And what's become clear, though, is that there's a lot more variation going on. So that what I talked to you about there was this kind of very large-scale kind of changes. But there's also changes that are much more subtle that we've been able to pull out in more recent years as the technology has developed. So, of course, what we know is that each of these chromosomes is actually a big, long string of DNA with A's, G's, C's, and T's. So it's one long linear sequence. For so quite a lot of the genes we're still trying to figure out how they work, but there's a type of gene called a protein coding gene where actually we know probably a great deal about how these things work. And this is based on a kind of a really amazing code that was cracked by everyone knows that Watson and Crick determined the structure of the double helix, and what an enormous, I mean, it was an amazing discovery, right? But less people know about this discovery, which was actually a series of discoveries that Francis Crick and others were involved with after the double helical structure was discovered, which was the question of how do these strings of A's, T's, G's, and C's code for proteins. And proteins are known to be strings of amino acids. And there are 20 different types of amino acids, and this shows you just the names of some of them, so such as leucine, serine. Each of these amino acids, although they can be strung together, each of these amino acids has a different property because they have different side chains. So some might be positively charged, some might be hydrophobic, some might be negatively charged, and so on. And the problem was then, how does this four-letter code somehow turn into, in DNA, A's, G's, C's, and T's, somehow turn into a 20 amino acid code? And what Crick and others figured out was that there's a triplet code. And they gradually decoded all these different triplets so that you can see that 3 use codes for phenylalanine. Oh, sorry, I should explain. The reason that these are U's rather than T's, it will become apparent in a minute, so don't worry about that. But basically, a U is the same as a T for this purpose. So I'll come back to this later, but this is a really, really important discovery that, in fact, holds across the whole of life. So in all different organisms on the Earth, they're using this kind of code. So this is really the code of life. And I'm always amazed when I see this and think about it. So this is how it works. If you have a protein coding gene, this is a beautifully complicated picture of the double helix, 2 strands of DNA. One of those strands of DNA contains the information for the transcript, which then, hopefully, there you go, which gets read off in an intermediate form called messenger RNA. So that's just a copy of one strand of the DNA. And as I said, I wanted to explain to you that instead of putting T's in there, it puts uracils U's in. But other than that, it's completely the same. It's gone ahead of itself, sorry. And then the protein reading machinery comes along and reads off this transcript and inserts the appropriate amino acid at each point. So you end up with this string of different amino acids, methionine, glutamine, glutamic acid, and so on. Why does that matter? Well, remember I said that the amino acids all have different side chains, so they have different properties. So you can make these different combinations, linear combinations, and the particular combination of amino acids that you have determines the shape, the way that this protein folds up. And the key thing here then is that the way that the protein folds up determines how it fits with other bits of machinery in the cell with other proteins. You can make a potentially limitless number of different combinations. Linguists pay attention to this. You can get its discrete infinity. These different proteins can do all sorts of different jobs in the cell. They do jobs like, they can be channels, they can be enzymes, they can be structural proteins, and so on. And you end up, in this way, the genome is able to code in a linear form the sort of machinery of life, which again is an incredible thing to think about. So DNA goes to this intermediate message, which is used to read off eventually, which is used to read off to build a polypeptide, a string of amino acids. This folds up in a certain way. And these kind of protein codes is what we're interested in for studying how these kind of genes work. So what is the impact of these proteins and how do they work in the cells and in the body? Now, let's get back to variation. So what it turns out is that, of course, we can have variations in these codes. And sometimes these variations will not have any impact because it's a redundant code. So what I mean by that is that in some cases, for example, if you look here at this leucine amino acid, it can be coded by all these 4 different triplet codes here and also 2 here. So you can imagine if you had a mutation at a certain point and it changed it in a certain way, say in the third base of this triplet, that actually it won't make any difference for the protein that's being made. So here this shows you this type of change. So here this glutamic acid here, if you change the third G to an A, that mutation leaves the protein doing what it always did. But let's look at this again and say, well, what would happen if you were carrying around a mutation here, a G to an A? At this position, it actually changes this glutamic acid to a lysine. And this could change the shape of this protein that this gene is making and actually give you a problem because the protein might not work as efficiently, or it may not work at all, for example, because of this change in shape. Now, at that same position, you might have another change which would actually turn these amino acid codes into, there's 3 codes that say, stop, this is the end of the protein, stop making it, otherwise these things would just go on forever. Right? So this is where to stop. Now, if a stop gets inserted in the middle of a protein, you can end up with a truncated protein that doesn't work at all, for example, or that could have extra properties that it wouldn't have normally. Now, there are lots of these variations again, sitting around in the genome in all of us, and we've, in recent years, we've become aware that there's actually a lot more of these kind of damaging changes that we somehow walk around with and cope with. But some of them, these things cause disorder. Yes. So actually, so some of you will know this. This is of the esteemed neuroscientist Scarlett Johansson, and it's been reported recently in the news. She's recently rediscovered this fact that all neuroscientists know, which is that we only use 10% of our brains. So if only we could increase the amount, then maybe we could become spies or what. I haven't seen the film, so I don't know. But actually, Scarlett Johansson missed a trick here because it's much more interesting what goes on in the genome because we only use 2% of our genomes. So that's a bit strange, right? So if you can increase the amount of genome that you're using, then please do try because it could really help. There's a serious side to this. So it was a big puzzle when... Well, there you go. Oh, magic numbers. So the point here is that most of the genome, for a long time, most of the genome, we didn't really understand what it was doing. And the protein coding genes that I talked about make up only 2% of the genome. So it's a very small percentage. So there's lots of other sequences that we don't know how they work, and we're beginning to figure out that they do. A lot of it does have sort of regulatory functions that maybe we'll hear more about in the course later on. But some of the things we do know how they impact on protein function. So here's one example. So the first point here is that in front of every gene and also kind of around the genes, there are regulatory sequences that govern how that gene is being switched on and off. So you might have 20,000 different genes, protein coding genes in your genome, taking up 2% of your genome. Those 20,000 proteins can't all be switched on at the same time in all your cells because then you just have a broken machine. So different cells have to have different profiles of proteins being switched on and off. They have to respond to environmental cues. They have to respond to signals from other cells and so on. How this works is that there are sequences in front of the gene and also, so there's a particular sequence called a promoter that sits at the front of every gene, and it tells, in concert with other things, it tells the gene whether it should be switched on or off. And then there's this weird fact that most genes are interrupted by these things called introns. So the exons are the part that code for the protein, and then there are these introns that the cellular machinery has a whole special load of factors that are able to cut out these kind of intronic bits, like editing out bits of piecing a movie together in the right order, taking out redundant bits. So we don't know why this. Well, maybe I'll get back to why this might be important. Yes.
What triggers the promoter?
Something sticks to it, basically, a protein. So I'm going to get back. There are regulatory, as well as these proteins that are the sort of enzymes in the machinery that works in the cell. There are proteins that their job is to regulate how other genes are being switched on or off. So there's a whole suites of proteins that sort of act at the promoter to recruit the transcriptional machinery, which is also involves proteins, to this place. And so by sort of strengthening or weakening those kind of interactions, you can, this impacts on whether a gene's being switched on or off. And I'll come back to that a bit later. So after a gene is, after a messenger RNA transcript is spliced, there's also the point that not the whole of the transcript gets used to code for the protein. As I said, there's a kind of stop sequence at the end. There's also a start codon as well, which is AUG, which codes for methionine. So methionine is the first amino acid that gets included in a lot of these proteins. Sometimes it gets cleaved off in the mature protein, but it's used as the kind of start sequence. So there's stuff at this end of the gene that doesn't go into the protein. There's stuff down here, and they also have these little tails called poly-A tracts, which you don't need to worry about. But the pointer here is, and that's what gets translated into protein. But the key point here is that what I talked about before were changes that could potentially change the amino acid sequence of the protein. But there could be changes that do not do that because they're outside of the protein coding region. But they could still have an effect. So for example, if you had some variation here at the promoter, it might stop how well some kind of factor that's coming in to switch on this gene, how well that sticks to this part of the promoter. So that might actually reduce the amount of transcription from that gene. So there could be variations in the promoter that could have subtle effects on how much, or actually even major effect on how much of that gene is being made. It doesn't change the shape of the protein, but it might change the amount of the protein or the way that the protein's being switched on or off during development. Similarly, you might have sequences that that affect this process of splicing. So they may not be in the protein coding regions, but they may be in the introns or near to the boundary where it's supposed to be cut out, and they could affect how well the splicing machinery is cutting out these bits. And if you can imagine that then this might affect this process and might actually again reduce the amount or even sometimes change the protein that's being made. Because if the protein making machinery starts reading into an intron, it may just start reading out garbage, sort of weird amino acid sequences that shouldn't be there. And then it's become clear that there are regions in these messenger RNA molecules that affect how stable they are or how well this process happens, whether they're actually used to make lots of protein or not. And so these kind of level of changes can also have impact on the amount of proteins that should be made. Now, another point I just want to make is to go back to one of the things about having introns, having genes interrupted, is that we don't know if this is the reason why we have them, but it gives an extra level of flexibility because many genes show something called alternative splicing. So it could be that in some versions of this gene, this exon gets stuck straight to this exon, and this one gets left out. And if that codes for a particular domain, a particular part of the protein, then you can make a different version of the protein in the same person that is doing different things, or slightly different things. So this means that even though you've got 20,000, about 20,000 protein coding genes, you can make a great deal more proteins than that just by this process of alternative splicing. And it seems to be there's some very interesting pathways in brain development that depend on this, which I won't go into now. But, so just to summarize the sort of this issue about variation, just to emphasize that there's this enormous amount of silent and functional variation in the genomes of all of us that we're only just being able to see with new technologies. And we've known about quite a lot of it for a while, but we're seeing more and more of it. And it is of these different types. So I've talked about chromosomal rearrangements. So these are these big, large-scale, fairly rare changes where you have translocations, deletions, duplications, and inversions that you can see under a microscope. In recent years, it's become clear that there are actually also smaller versions of these kind of deletions and duplications, which we haven't been able to see before, with sections that are where genes are being duplicated or being deleted. And these are actually just really small. They're just small things that we knew about these things before. They're just small deletions and duplications, but they've been given a new kind of name, which is copy number variants, CNVs. So you might hear a lot about these in relation to diagnosis of disorders, because people have got interested in how these kind of copy number variations we're able to detect very easily now, how those impact on if you have an extra copy of a gene, perhaps you make more of that protein than protein that it codes for, or if you've got a lower copy, you might make less. So I'll talk maybe a bit more about that later. But and then we also have at another level, we have another sort of grain. We actually have just individual changes to individual bases that might just change one base from one DNA letter to another, or sometimes insert just a very small number of changes. So these are very tiny changes at the genetic level, but they can have really big consequences if they might change the sequence of a protein, or insert a stop signal, or this is a thing where another type of change that messes with the sequence of the protein. And I've said to you that even if you have changes that lie outside of the protein coding regions, these could still have functional impacts, because they might affect these other processes that regulate how much of a particular protein is being made in a cell. So splicing, transcription, translation, and the stability of transcripts. And importantly, there's a whole kind of massive wide range of different frequencies for these things. So sometimes there are variants that if I just took a particular variant at one point of a gene and tested it in all of you, 50% of you would be carrying this variant. And Dorothy was talking about these kind of variants yesterday, and these are kind of, and also Mabel, I think, was talking about these kind of variants on, I can't remember which, Tuesday, Monday, lost track of time. But these are called single nucleotide polymorphisms, and the reason they're called polymorphisms is because once it's at a certain frequency in the general population, it's considered to be polymorphism as opposed to mutation. But these are all, you don't really need to worry too much about the terminology. The point is these are variants that can occur either widely in the population, but the flip side is that you might sometimes see variants that are private and just found in this one individual or this one family, and you see everything in between. Okay, so there's a whole host of different, every 20%, 30%, and so on. And it's a situation that will be in flux as we kind of, as the human population goes on, but you just have to bear in mind that we have all this variation, rare and common variation, that is being carefully documented.
So a question.
Yes.
So all these kinds of changes, are they random, or there's some kinds that are much more likely than others? So how are they distributed, and what percent of them aren't even compatible with life in the first?
Yes, that's a whole other talk, but yes. There's a range. There are big questions about whether there are certain parts of the genome that are mutation hotspots. So, for example, for copy number variants, there are regions which are particularly susceptible to copy number variation because they have repeated sections, and this makes them more unstable. So there are mechanisms in certain places. If a gene's really big, obviously it's more likely to have mutations and variations in it. Some genes are under higher constraints than others, so that some genes really don't tolerate, if you have a mutation, a functional mutation in them that knocks them out, it's incompatible with life. Some genes are more redundant. Sometimes that's because also you have other copies, or, well, you have gene families sometimes, and it can be that they're working together, so that if you lose one gene that's doing the job, the other gene somehow sort of compensates to a certain extent, so it's still compatible with life. But there's a huge... so the Human Genome Sequencing Project was about getting the first... I'll come back to this, about getting to the first human genome sequence. And now there's the data that's accumulated over the past few years has really been focused on documenting the shape of the variation, and also the shape of it. I hope you'll hear about this later in the course, also how things differ from other species as well. But these questions are really interesting. Sorry.
Well, the percentage of redundancy in the genome is there.
I don't know. I don't know if anyone's actually sort of.
Wouldn't the redundancy change depending on the environment as well?
Well, that's also a point, yes. But
What about the percentage of knockouts that show no effect? That's a number that we know, right? Since virtually every gene has been knocked out in mice, and isn't it like half of them basically you can't tell?
yeah, a lot of them you can't tell. But I don't know if we've knocked out. Yeah, I don't know how meaningful these kind of questions are, but actually, because it's a kind of a lot of it's about a statistical description of how our genome is at the moment, and how it varies. I think the really interesting questions are how these variations relate to phenotypes, and well, I guess and evolutionary change as well. But so they're probably answers to your questions, but I don't know them off the top of my head. Sorry, Lena.
So Williams syndrome, is
Yes.
it known whether that's in some kind of mutation hotspot?
It is, yes, because it has these regions that flank it, that have been very well described. And what's interesting about Williams syndrome is that Williams syndrome was known for a long time, and studied in some detail. And then everybody knew that there should be, for most of these syndromes where there's a bit deleted, there is also a duplication syndrome, because the same mechanism that makes this area prone to be deleted makes it prone to be duplicated as well. So people kind of knew about this thing, and then actually in, I think, 2005 or something, Carolyn Mervis and Lucy Osborne reported the first case, and now there's a bunch of different cases of duplication of the same interval as Williams syndrome interval. But so I'm not so concerned with describing the specifics of how much variation there is in these kind of things. But all I want to really do is get at the idea that there is a lot of this variation around, and that we can tap into it and try and relate it to variation in language development, and in pathologies, but also maybe in normal variation. But we'll get to that later. And the point here is that a lot of these variants can have a silent and won't have an effect at the kind of cellular machinery level. And in that case, you might not expect them to do anything. And then you have some that are severe, and it's often actually that the rarer ones have the severest effects, but well, we'll see how that turns out. So then we get back to this question of finding some kind of a level at which we can study the variations, and I happen to consider that studying pathology is part of this kind of thing, but I've noticed in say discussions with Chomsky, he says, well, that's pathology, that's not kind of really very interesting, and that there isn't any actual variation, say, in the language faculty in all of modern humans. But I would consider that pathology and normal language variation, you can apply all these same approaches to. But I'm going to talk more about pathology, I think, today. So the idea, again, is that we now have a lot of powerful tools for getting at this level. So and the tools have changed really, really dramatically over the past 5-10 years. I mean, really, I'm astonished by what's happening now compared to what was happening, say, when I was doing my PhD. So this was kind of the situation for a while, which was in terms of the tools that we had available to us. And it went a bit like this. And I think that now we're at this kind of stage. So there's a lot of different things that we can do, and a lot of tools for capturing genetic information in the people that you are studying. But one of the points here is that these tools are not enough, right? I'm going to go over some of these tools, but then we'll get back to using the tools responsibly at the end. So when we're talking about the level of sort of chromosomal abnormalities, the breakages, translocations and deletions and so on, we used to have to see big things from these karyotypes where you could just look under a microscope at somebody's chromosomes and try and see what's going on. And I'm not sure if this was like 10 years ago or something like that, I'm not sure, maybe it was longer ago. But people started developing these ways of actually breaking up DNA and studying it on arrays. So you might have this shows you an example where you have a patient who has doesn't matter what disorder they have, but they have a particular trait, and then you have a control DNA, and you basically are labeling the DNA, cutting it up, labeling it, and sticking it to a kind of array, a sort of chip where you have lots and lots of fragments of the genome basically positioned all along it. So you can see across the whole of the genome, you have probes for different sections of the genomes. So you can sort of measure how much green there is and how much red there is. And basically all this tells you is if somebody has, you can kind of read it off here, how much the green signal is versus the red signal. And your argument would be if there's a difference, if somebody had a duplication of a region or a deletion of a region, then you would see more or less sticking on here and you would see different color. And it's kind of I hope it's a simple idea to understand, but this just gets at a new way of thinking about this using these arrays led to people being able to screen people where they'd only before been able to do this kind of karyotyping approach. Say you had a kid with intellectual disability or autism or something like that and they came into a clinic, you would be able to look at a fairly crude level at what was going on in the chromosomes, and if you didn't see anything, then you didn't have much else, another place to go to. And when these kind of arrays came in, people could start to see these copy number variations, and it led to a whole host of new discoveries about all the extent of variation that we actually carry around. And now there's big questions about how these things contribute to pathology. So that's really changed the field a lot. Now, the old way that we used to, of course, that's just looking at how much we have of each thing. But what you really want to be able to do is look at how these individual sequences vary in people. And we used to not be able to see that variation very well. And we used to use these kind of techniques where we would use kind of signposts in different parts of the genome where we knew that the DNA varied in some way. So for example, if you had, there were stretches where there would be particular sequences that would duplicated, not coding for genes, these sequences. And then that would make them unstable. And so some people would carry one length of this thing, and other people would carry another length of these things. And you can see here on a gel, this is, I'll tell you about this in a minute, but this is a family with a particular disorder, and this is a probe that's being used to pull out variation on a particular chromosome, actually on the X chromosome. And this probe is called M27beta, and you can see that this woman carries these two versions of this probe here, this marker. Actually, that doesn't look right now, come to think of it, because, hold on, I'm not sure how these match up to these different people, because I was just thinking there's a male with two, which can't be right. But anyway, the point is that maybe this is this guy here. The point is that we can see these variations in people, and we use it as a kind of signpost to say this tells you how this section of the chromosome is being passed on to different people. And I'll come back to this later so that you can understand it better. But the point here is that this kind of stuff that we used to have to do was quite laborious, and would take us a long time to look at just one marker in one person. So that you could spend weeks just studying one family for one marker. So that was quite a lot of work. This actually comes from a family with a kidney stone disorder. I discovered a gene for kidney stones when I was doing my PhD. And it's interesting because when you work on kidneys, you never get somebody saying, it's the gene for kidneys. Or anything like that. But in language and other places, it goes a bit different. So then genotyping evolved to an extent where we could do more than one marker quite quickly. And this is where these are gels that are similar to these last gels, but they're colored, and the red lines are just kind of invariant. They're just sort of a way of marking the gel. But each of these coloured bits here is a different genetic marker that we're testing, if you like, in a family. And each of these lanes is one person. So here, I don't know how many, there's 40 or so people that we're testing on one gel, and we're testing 20 markers across from different parts of the chromosomes on here. And some of us spent many happy months just generating these kind of things and you run them, and then a laser scans and creates this image, and it gets converted into something that looks like this for each marker. And what you can see here is a typical family where there's mum who has she's got two this is again this is not in a gene per se, this is just a marker of a particular chromosome section. It's a bit of a chromosome that varies, and we can see how it's varying. So the mother, she has this, don't worry about these bumps that come up to it, but this peak here tells you that she carries one version of this gene here and one version of this gene here. The father carries this version and this version. You can see that this is the child, the first child that they've had has inherited this from the father and this from the mother. This sibling has inherited this one from the father and this from the mother. So actually for this marker, these siblings are completely different. They've inherited different variants. So you can trace the sort of how the chromosomes are being passed in families. And then you can see here that both these siblings are actually getting the same variant, this variant from the mother, this from the father. So this was very laborious. And we used to call this semi-automated because these things got generated automatically and then we had to check them through by hand to make sure that the programs were doing it right. Now what people do, instead of doing this laborious stuff, is that they realize that we could use, remember, everybody has these single nucleotide changes. So if we know enough of those single nucleotide changes that are common, we could just look for those and see whether you carry. So if we say that at a certain point in the genome, 50% of people will carry a G and 50% won't, then we can design a probe that tests whether it's a G or an A just at that point. These get arrayed on these chips again, and then you can simultaneously, in one single person with one of these chips, these are actually very old, the chips now I think are a bit better than this, but probably a bit smaller. But they allow for you to test hundreds of thousands of polymorphisms across the whole genome. So you can take a single person and in an afternoon you've got a million different points of their genome. So that is a massive change. So another level that we can go to is, because what's that doing is looking at known signposts that we already know about and recapturing those in the person. But what about if somebody carries something that's, I remember I said some people have private variations. How would we ever find those kind of private variations? And then you need to actually sequence them to find them. And this is how we used to do sequencing. We used to run these very laborious kind of gels, which we could read off manually. And I'll show you how. So here is each of these tracks tells you whether there's a C, a G, a T, or an A. And you start at the bottom and you go C, 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 G, C, T, G, G, and so on. So much fun is had by all. I did quite a lot of this in my PhD. And then we were very excited when these kind of fluorescent technologies that I showed you before, where you could run a gel with lots of pooled things and you can automate the kind of reading of it. When this came along, there were ways we moved to these kind of electropherograms where you can see, you can just read from left to right, A, G, A, and the computer normally gets this right and you don't need to double check it, although it's worth double checking that you haven't got problems when you see changes like that. But this actually, the problem with this kind of technology was you were still sequencing little chunks of the genome individually as you'd go, I'm interested in this section of the genome, so I make lots of it using a clever technique called PCR, and then I sequence that bit. And that's how most of the human genome was initially decoded. Now what's happened in the past few years is that sequencing has evolved to a completely different level by people thinking kind of in parallel ways. So they use kind of platforms where you can sequence you fragment the genome into lots and lots of pieces, and then you sequence all of it simultaneously in different ways on chips and things like that. And what this means, and here's quite an old machine now actually, but this kind of machine can do millions and millions of sequence reads in just one run. So remember I said to you that you can take that chip before and you can get polymorphism. You can test polymorphisms at hundreds of thousands of points for about a couple of €100. Well, these things are doing just sequencing the whole genome, or large chunks of it. Now, the initial human genome sequence was about $3 billion, I think. It took lots of scientists over 10 years to complete. I was one of them involved with this. By 2010, because of these new kind of technologies, a human genome could be sequenced in a month for less than $30,000. I'm not sure, but I think that I think we can currently get a deal of about $2,000 for a genome now. It depends how well you negotiate. But so there are companies now who will do this. That you can take somebody spit into a tube, you send it to a company, and they will send you back their genome. So we are now in the era of personal genomics, well into it actually. We've been in it for a while, and almost like consumer genomics as well, because people can send their DNAs off themselves if they want to, and if they can afford it. And what I want to argue and show with the last sort of half of the talk is that that's not enough. And it's very seductive for young people nowadays because they come into the field and they go, hey, I want to do genetics because I can get these tubes that I can buy from wherever, and I can send them off, and it's all. And I would say I would encourage people to do this. I think it's really that you should think about using genetics, but use it in a clever way. So the genetic architecture, so going back to speech and language, which is one of the things that we're here to talk about, there's no question, based on everything else that we know about human biology, that the architecture underlying this is going to be really complicated, and it will certainly involve different genes with distinct modes of action. Some might have larger effects than others, we don't know. I've already told you that every human carries a large number of genomic variants that might be candidates for influencing phenotypes of interest. So it's still very hard then, when we know we've got a phenotype of interest and we have the whole genomic information from the people that we're interested in, we might see all sorts of relationships, and we need to find out which of those correlations that we see are real and which are biologically valid. And that's going to be a big challenge for the future, and there are ways of getting around it. And I think that you always want to think about genomics. It's very exciting, and I would never, as I said, I'm really amazed by the progress that's been made. These should be thought of as not the end points, but these are tools that you can use to generate data and to test hypotheses. So one way of doing this that we've already heard a bit about how this works, or how this has happened from Kate and others, is to find interesting multi-generational families. And I want to just show you why that's a kind of shortcut to finding interesting genes if you're lucky enough to get one of these families and it all pans out well. So if you imagine, here's actually a small family with, this is the mum and the dad, and they've had 2 children. And you can imagine, I'm just taking one chromosome to make this really simple, but the woman has this red and I don't know, cyan kind of chromosomes. So these are homologous chromosomes. They have the same genes aligned on them, but they might have different versions of the genes. And there's the dad. Now, they randomly might pass on to their kids. This is not going to work. Yeah. So in this case, again, this father has passed on the black chromosome to the son, and the green to the daughter, and the mum's passed on the red to both of them. And then these guys go out and meet and fall in love and have a nice big family, and some more chromosomes come in, right? And this, now what's interesting here is, this is just a family, everyday family, but what's interesting here is, you can imagine if this family had a speech and language disorder, for example, and these guys were affected and these were unaffected. You might say, well, maybe there's some kind of gene underlying it. And those of you who are paying attention will notice that something interesting is happening here, which is not at this black chromosome, because that doesn't co-segregate, or at this green chromosome, but this red chromosome is being passed on to all the affected people, but none of the unaffected people. So it's kind of co-segregating with the disorder. So and this might actually be because somewhere on this chromosome there's a mutation. It might be just private to this family. Who knows? But the point here is that it's on this chromosome, and that's what's causing the disorder, and that's what's causing the linkage. This is where linkage comes from, between this chromosome and the disorder and the trait. Now, that wouldn't be that much helpful, because then you just narrow things. If you did an analysis of their genomes, just looking at markers along the genes, you might be able to say it could be any gene on this whole chromosome, because they're all co-segregating with this trait. And luckily, nature has given us something called recombination, which is that sections of maternal and paternal chromosomes get exchanged during generation of germ cells. And what this means is that, and these, or we're talking again about mutational hotspots, so these kind of, there's debates about whether certain chromosomal regions are more inclined to be the sites of crossing over events. But the point is that as a result of this kind of shuffling around, this recombination of chromosomes, you end up as a kind of patchwork. You're a patchwork of all your grandparents' genomes all being kind of shuffled around. Now, geneticists can use this to track down in families like this where genes are. So here, if you look now, what I've done is make this family, I've kind of shuffled around all the chromosomes so that, like for example here, there was a recombination here, and there's a recombination here, and so on. So these guys are kind of patchworks of the chromosomes from these guys, and these guys as well. So now if you take a look at the red chromosome again in the affected individuals, you can actually see that there's a section of the red chromosome that co-segregates, not the whole of the chromosome, but there's a section of this original X chromosome, sorry, of this original red chromosome that is found in all the affected people, which is somewhere between here and here, and that's where the gene lies. Now, so this is how linkage can be used to track down genes of interest, and this is how we actually track down, initially how we track down FOXP2 by studying the K family. And I just want to say also that these kind of principles, now that we can sequence whole genomes, these principles of linkage still apply. So it's still the case that you can look in these multi-generational families to see that a mutation is actually linked to the trait, that it's actually co-segregating in a large family. And it might be that the same gene might have different mutations in different families, which is what makes it different from, say, what Dorothy was talking about yesterday, which is association across larger populations, where it's the same there in lots of different families or lots of different people. We've heard a lot about the KE family and the phenotype, which is great, so I don't need to go into any of that, which buys me some time. Does everybody get what I've said so far, roughly? Okay. So back to the socks. So what we were able to do by just that kind of fairly basic thing with the KE family of doing linkage analysis of different sections of chromosomes was we were able to zoom in onto a section of chromosome 7 and then sequence genes on chromosome 7. This was actually at a time when the human genome hadn't been finished, so we were actually sort of having to make use of individual sequencing data that we were generating to figure this stuff out. And we identified, this is work with Cecilia Lai in Tony Monaco's lab in Oxford in collaboration with Farina's team at Institute of Child Health. And we found that there's one of these very tiny changes, a G to an A transition in this gene called FOXP2, that is found in all the affected individuals of the family, but none of the unaffected individuals. They all have a normal copy of this gene. There's no changes in the gene. And we've also not seen this outside of this family. So this particular mutation is specific to this family, right? And it probably, we don't know, maybe it was, we know that the grandma was affected, so maybe she was the first person to inherit this and then she passed it on. Maybe there's, I don't know if there's any history in the great-grandparents.
She said she was dropped on her head.
Yeah, I don't think that would have done it. I don't think you can change FOXP2 by dropping someone else. But she's passed it on then to about half of her kids, who will then pass it on to half. And this would continue as, you know, through. But it's a private mutation to the family. This is what remember genes code for protein, so this is what the protein that this gene makes looks like. It's a very simplified image where I just sort of stretch the protein out. So one end we have something called the N-terminus, at the other end we have something called the C-terminus. You don't need to worry about why they're called that. And there's a few interesting domains, and the main domain that gives this gene and the protein its name is this FOX domain, fork head box domain. And we were lucky because this gene, when we discovered the gene, it was a new gene, but it belonged to a family of different genes that was already known, and they all have these kind of fork head box domains. And it was roughly, this is again a very crude picture of it, but I just find it appealing to look at. So this shows you kind of roughly what the structure of the DNA binding domain, this fork head box domain. I'll tell you more about it in a minute. This is what the structure looks like. So you have these 3 helices here, 1, 2, and 3. And this kind of domain in all these different FOX proteins likes to stick to DNA. And this section of the protein, this third helix, is the recognition helix that gets buried into one of the grooves of the DNA. The KE family have a mutation that is in the protein coding region that changes. It's called a missense mutation because it changes the amino acid from one amino acid to another. So this becomes, from an arginine, it becomes a histidine. We call it R553H because it's 553 amino acids from the start of the protein. What we know now is that if you stick a histidine in here instead of an arginine, it messes with the way that this domain can stick to DNA. And this is work that was done by Sonia over there. So
I'm actually going to show the experiment that proves.
yes, excellent. So you'll be hearing lots about this. So one of the copies of FOXP2 in this family has a problem with DNA binding and maybe some other problems as well, but it's dysfunctional. But they also have a normal function copy. So that's one of the mutations that we know about in FOXP2, but it's not the only mutation. And this is something that frustrates me slightly because people always talk about the K family and they have the FOXP2 gene and the mutation. And actually there are other mutations out there and probably quite a few that we don't know about yet. So the first one I want to talk about briefly is from an Australian family that we call the AD family. And they have one of these mutations that inserts a stop signal in the middle of the protein. So this changes, there should be an arginine at this point, and actually it just goes stop, don't make any more. We would never have found this if we were just looking at this family. There are here two kids initially diagnosed with developmental budespraxia of speech. There's lots of work going on now trying to compare these kids to what's being seen in the K family, which I won't talk about today because I don't have all the data yet. But the point is that they were given the same kind of diagnosis. The mum has a history of speech apraxia as well, and they have this mutation. And we only found this because we already knew to look at FOXP50 kids with speech apraxia or verbal dyspraxia, trying to match the phenotype that we were seeing in the K family, and we found this mutation, and so that was kind of the second of these kind of point mutations. I should say that there are other mutations out there. There's one I haven't got on here that's Angela Morgan's reported on in Australia. And you can see that gradually these other cases are emerging. But the point is, this is rare. This may explain like a small percentage of kids with speech dyspraxia, but it's a real effect. And we also have these cases of translocations that I talked about. So this translocation, this CS case, was actually one of the key things that helped us to get to this gene in the first place. So when we were studying the KE family and we knew to look on chromosome 7, we looked out for kids who had translocations in that region of chromosome 7. And Jane Hurst discovered this kid, and she was the first person to report on the KE family, actually. She discovered this kid with this translocation, and that's what actually got us zooming in on this region of the genome and pulling out this gene and finding FOXP2. And then we went to the KE family and found the mutation in the fork head box domain. And then Bruce Tomlin has a family with a translocation, and actually there's a few others now out there with translocations and deletions of various sizes. And so what we would say from this is that 2 functioning copies of FOXP2 are needed to acquire proficient spoken language, which I think is a fairly understated claim, but damage to one copy causes this disorder that you can argue about whether it's motoric or, you can argue what the origin of that is. But the point is this is an interesting, rare molecular entry point into these kind of pathways. And here's what FOXP2 does. You'll hear more about this later. So FOXP2 is a transcription factor. So something comes along and switches on FOXP2. It gets transcribed and spliced. And then a protein called FOXP2 is made. And FOXP2 goes back into the nucleus where it likes to find other genes. And it might bind to regulatory region of target gene X and switch it on, activate it. Or target gene Y, repress it. So, and what we actually think is it doesn't happen really like this. It's not an all or nothing thing. It's kind of like a tuner, like a genetic dimmer switch. I used to say, I don't say this anymore, I used to say that mostly FOXP2 acts like this, but Sonia's been finding lots of evidence of it acting like this, so I'm not allowed to say that anymore. So. But the point is that these are kind of tuning effects. They're not like, oh, FOXP2's on and everything else gets. So just bear that in mind. And now I'm going to just briefly, Sonia will return to this, I think, in her talk, but I'll just briefly introduce this idea that if we know about a particular, although FOXP2 mutations are rare, the nice thing about it is it gets us into a piece of biology that we can then pull apart and try and go back and link it back to language or speech or any related phenotypes. So it gives us a kind of window into, we always talk about it as a kind of molecular window. And it's particularly nice that it's a transcription factor because we can ask about what are the other elements of the pathway that it's in. What are the things that it's regulating, for example. And Sonia found one of the first genes that it was regulating, which is a gene that was known at the time that I'm going to call catnap2, because that's what we call it. And this is, we've actually heard a bit about this from Mabel already. So this happens to be a pain of a gene to study, I have to say, because it's the biggest, now it's the biggest gene in the genome. For a while it was the second biggest, and now it turns out to be the biggest. I don't know what happened, but went up in the charts. So it's about 2 million bases long. That's very big. And the reason it's so big is because it has lots of big introns in it. Remember, you have exons and introns. The diamonds are exons. And what Sonia showed with some very clever techniques that I won't go into was that one of the places that FOXP2 is sticking in the genome is over here, and it's a regulatory part of this catnap2 gene. And she went further to show that if you mess around with the levels of FOXP2 in cells, then this would lead to downregulation of catnap2. So there was a correlation where sort of an inverse correlation, so that if you increase levels of FOXP2, then catnap2 levels go down. And this, we seem to see this a lot in different experiments. And Dan Geschwind and Brett Abrahams in UCLA did some nice work where they actually labeled in human mid-gestation fetal brain tissue. They could label where catnap2 and FOXP2 were switched on in this kind of laminar structure of the developing brain. And they, again, you can see a kind of inverse relationship. You're going to hear, I think, a bit more later about where FOXP2 switched on, but it tends to be expressed in the deepest layers of the cortex during early development, whereas catnap2 is much more superficial. So again, there's a kind of inverse correlation, a bit like what we were seeing in the cells. So we think that's kind of an interesting molecular link. You will hear more about why this catnap2 gene is particularly interesting in brain development, so I'm not going to spoil that for you. That will be in Sonia's talk. Yes?
When you say so superficial layers, that's
Two,
2, 3,
3,
5, 6 is, and that's
5, 6 is FOXP2. 3, 4 is Fox. Oh, no, I'm going to spoil my next bit, so I won't say that. But anyway, there's an inverse relationship between FOXPs and catnap2.
yeah, but then how specific can you associate them with specific layers?
Yeah.
Deep and so
Yeah. So FOXP2 is primarily layer 6. I don't know, maybe also Wolfie might talk about it. But FOXP2 is primarily layer 6 and maybe a little bit of layer 5. It varies a bit. Less is known in the human brain. Much more is known by studying mouse brains. Yeah.
FOXP2 is very commonly a layer marker range.
It gets used as
basic anatomy. People using different models.
yes. Matt.
And is it entirely cortical? It's not expressed?
No. It's not entirely cortical at all. No, it's also expressed in lots of other places. But I don't.
Kate's straight or finding?
Yes, it's. Okay. Well, it's highly expressed in the striatum. Yeah, it's one of the key sites is the median spinal neurons of the striatum. It's expressed in a. I don't want to get too much into that because I don't want to spoil Sonia's talk. But it's not expressed all over the brain, and it's not expressed just in one spot. So this result is just showing you what goes on in the cortex, but you'll hear more about what it might be doing in other places. It's also not specific to the brain, which you'll also hear about later. So then, so this kind of highlighted this might be an interesting gene, and Diane Newbury from Oxford, who's been leading this with Tony Monaco, who's been leading this specific language impairment consortium, which Dorothy's also involved with for some years now. She had 184 families, not with the sort of FOXP2 type of cases of these speech dyspraxia, but kids with typical diagnosis of SLI according to the standard criteria that people use in the UK at the moment. And so they have common forms of language impairment. We excluded kids with autism from this study. And as I think Dorothy said yesterday, this was being used to study various different candidate genes. But this looked like a very nice candidate gene based on the link to FOXP2. So Diane tested a series of markers. And now these are common variants. These aren't rare mutations. These are common variations again. And found association in a section here with a particular set of SNPs. So the kids who carried this set of SNPs within these SLI families, these small SLI families, got worse scores on language-based tests. And the most dramatic findings were seen for non-word repetition. Now, I don't know why, but that's what we saw. So we did also see with more basic expressive language, maybe a little bit with the receptive language tests. We use these kind of something called the KELF or the SELF, which is a battery of language tests. But the most clear-cut relationship was with non-word rep. But it's now I'm wary of the fact that you said, always report effect sizes. I don't have a slide with the effect size, but it's a small effect size, again. So it's not like it explains the whole of SLI by any means. But if you carry this variant, then, or in this family set, if they carried this variant, then they did worse, and the effect was significant after adjusting for the markers that we tested. Dan Geschwin had also been interested in this gene for various reasons, and he was studying autistic children, but he wasn't studying them saying, are they autistic or not? He was getting data on age at first word from the parents and using that to test for association. And he independently found, and these, again, remember, these families didn't have autism. We excluded anyone with autism. These are kids with autism and a measure of just simply of age at first word, and he found that the same markers were associated. And this was actually an independent study. So we kind of converged on the same markers, which I thought was quite interesting.
When you say the same marker, you mean the same SNP?
Same SNPs. This set of SNPs here. And same direction as well, which means that well, not the same direction because it's not the same measure, but the things that were, the alleles that give you reduced non-word repetition in the SLI sample were the same ones that were correlated with later age at first word. Sorry.
So this is common variation of CatNat2. What about common variation of FOXP2? Do you see that?
You don't, and I will return to that at the end of the talk. But so what we haven't seen really convincing evidence yet. There's a few. I'll show you some data right at the end on that. So then, and actually that was something that. So one of the first things, obviously, that we did when we found FOXP2 was to go into these families, which was a smaller number at the time, and study FOXP2 variation in those families, and we didn't see anything, and so we reported that we thought it wasn't likely to be involved in common forms of SLI. So that's interesting then that two different parts of a pathway are one involved in the sort of only in the rare, and the other involved in common and maybe also rare, but you'll hear more about that. And then we collaborated with Andrew Whitehouse and also Dorothy on Andrew works on this RAIN sample, a kind of epidemiological sample in Australia with, I think we studied about 1,500 kids in this study. I can't remember exactly, something like that. So it's a fairly large sample of kids. We studied the same alleles again, and we found that kids who carried, in this general population sample, kids who carried these variants were, on a parental measure of early language, were, again, worse off, if you like. Now, but again, I want to be quite cautious about the claims here, in that we see these associations, but they don't explain a lot of the variants, and they're on the edge of significance. But what's interesting here is that you have two different genes with distinct types of genetic effects. So you have FOXP2, you have rare coding mutations that change the sequence of the encoded protein, causing a monogenic speech and language disorder, and we haven't really seen clear evidence of any common variation being relevant, which I will come back to. And then in CatNat2, we have common variants that are outside the protein coding regions. That was interesting. But you'll hear more about CatNat2 because there's also evidence of rare variation in CatNat2 as well.
Sorry, how can we decide to choose the risk variant? Is it a minor allele or a measure based on the literature?
Well, we were just using standard SNPs. So we were doing a screen across, again, so we were kind of took the SNPs that were known across that gene at the time that were being used to genotype that gene. And we look at both, so we capture both alleles. So then the question is, we don't know which allele is going to be the risk allele or not. But as it happens, I can't remember, I think it's the minor allele in this case. But and the claim here is not that these are the variants that are causing it, because of course we're just using them as kind of signposts to the real variation that might be causing it. It might be another variation that we haven't tested or that hasn't been found yet, or that. So this is, maybe I'll get back to that right at the end. But the interesting thing here is that these things converge on a shared functional pathway, and that we see something going across different types of disorders. This kind of thing is coming up a lot these days, in that many of the genes that are involved in, or are claimed to be involved in things like autism and schizophrenia and dyslexia and so on, maybe less so for dyslexia, a lot of them are kind of being going across these disorders, which makes it hard for us to understand where the specificity comes from. But maybe that's what Dorothy was sort of hinting at. So I've given you this example of CatNat2. I'm not going to go into these other examples because you'll hear about them later. But this is this general principle of looking at downstream targets that FOXP2 regulates. But we should also, I'm just going to keep going. We should also, is it at your end or? No. We should also be studying other parts of the pathway, right? So we know that there's downstream targets that we can look at, but also we should try and understand the things that are driving FOXP2 to be expressed itself, the things that there are kind of signals that come into the cell from outside that might affect how FOXP2 is working, but also these proteins, these kind of transcription factors, in fact any protein at all, never acts by itself. It interacts with other things, and that's how it. So if you can find the things that it's interacting with, you might be able to study, you might be able to get another kind of hook, not just the downstream targets in the case of FOXP2, but the other things that it sticks to. So I was going to give you an example of one of these before I finished.
So just to follow up on the CatNat variant, so the causative variant is not yet known?
Not really, no. I mean, I don't think, there's no function, all we know is that we have something that's associated, and that we use as a kind of, so something, but at a cellular level, I don't think we have any information.
I mean, most of the variants are intronic, so not predicted to change the protein sequence at all.
So presumably it's something to do with how this gene's being regulated, maybe that more of it's made.
The haplotypes are not in linkage with any amino acid variant, right?
No, that's right, yeah. So it's not, I think we know that it's not an amino acid, whatever it is, it's not a protein change, but it could be something that's about how the thing's regulated.
And most of the associations were in this one area you showed, right? I mean, it was not that the normal language onset was at a different region.
No. So the three things that, all the stuff that I showed you there was the same regions, which was what got,
It's a little cluster of SNPs.
yeah. But that's not to say that there aren't other studies that have seen association with other. So there was originally a claim that there was something up near the promoter of CatNat2 that was associated with autism. And then a recent study looked in quite a large sample of kids with autism and said, CatNat2 is not involved in autism because they didn't see an increase in affected versus unaffected, right? But that's not really relevant to Dan Geschwin's finding, because that was about, in kids with autism, if you carry this variant, you do worse on language. That's the idea behind it.
When you find an association in infected individuals, can you then sequence the whole gene and see if
Yeah, that's what you should do if you've got enough money. But then, I mean, I don't know if Sylvia will talk about it. Sylvia's done some nice work on trying to figure out in this KIAA gene that's associated with dyslexia, actually what are the variants, where are they, and how do they act. So maybe I don't know, will you talk about that? Yeah, great. So you'll get an example of how you can do that. Let's see how far I can go with this before. Okay, good. But that's not good to know, really. Okay, let's see before it. So I want to give you an example from. I mean, you already know what autism is, but what I want to hopefully explain to you. So this is rationale behind a study that's now there's a lot of this kind of stuff going on, but this was at the time, a few years ago, Evan Eichler was one of the first people to sequence whole exomes of people with autism. So there what you're doing is using next generation sequencing techniques to determine the sequence of all the protein coding regions in the genome. Remember, Scarlett Johansson taught us that 2% of the genome is important and the rest of it doesn't matter. So we just sequence those 2%. So that's the exomes, and it gets you all the protein coding variation. And that was the reason for doing exomes was also because it used to be cheaper than doing whole genomes. And the reason that I'm kind of talking about autism here is to go back to this idea of sequencing the whole of the exome in kids with autism. But there's a trick here, which is that the idea here is that if you had very, very severe disorder like this, you might not then be able to pass on your genes to other people, right? So there would be a kind of negative selection against certain types of deleterious variants. So that led to this idea that in a certain number of cases of autism, these might be explained by de novo new mutations that have popped up in this kid that aren't kind of inherited, that have just been mutations in the germline of the parents, and then these things disappear out of the population. So there might be an excess of these kind of de novo mutations causing autism. So the Simons Foundation set up a cohort where they were looking for children with what we say sporadic autism. What we mean by this is not that autism kind of comes and goes, but that that it's just in the family. There's just a single case of autism. So it looks like this: a kid has autism, his parents are fine, the children are fine, the siblings are fine. So the idea then would be if you could sequence this guy's exome, all the protein coding regions, and you sequence the parents, then and you just look for new changes that are just in here and not in the parents and also not in the siblings, that actually narrows the search space a lot. So you can, because every generation there's a very small number of new mutations that are occurring in protein coding regions that affect protein coding. And so Evan Eichler and Brian O'Rourke at the University of Washington in Seattle were studying. They studied only about 20 of these families. Now lots of these families have been studied, but they studied 20 of these families, and they found a few different interesting mutations. And here's one of them that they found. It's in a gene called FOXP1. This kid has a point mutation that leads to a kind of a frame shift and a truncation. And it was novel, a de novo mutation, and it's predicted to kind of cut FOXP1 off in half. So it's a de novo mutation of FOXP1 gene that disturbs the sequence of the encoded protein in a kid with quite severe autism, including speech and language deficits, but also other stuff going on as well. Now, we were chatting with Evan about some of these results, and we thought, well, FOXP1, that sounds a bit familiar. I don't know quite why. So it turns out that this is the most similar gene in the genome to FOXP2 in humans. And like FOXP2, it encodes a protein which regulates transcription of other genes. In some parts of the brain, I don't know whether will you talk about FOXP1? Not really. But in some parts of the brain, FOXP1 and FOXP2 are switched on together, and other places they're not. But say, for example, in the striatum, they're expressed in the same cells. Hey, yes, excellent. So you'll hear all about FOXPs from Constance. And these proteins can directly interact with each other. So that's very interesting. FOXP2 actually likes to stick to another FOXP2 molecule. I didn't say this, but it sticks to another FOXP2 molecule to be able to regulate genes. Now, in places where FOXP2 is expressed together with FOXP1, you can get these dimers, these heterodimers, where FOXP2 is stuck to FOXP1. So that's very interesting. They can directly interact with each other, and so they might work together to regulate target genes in some of the places where they're co-expressed in the brain. There are some places they're co-expressed, some places they aren't. Here's FOXP2, and here's FOXP1, and they share this stretch here that's important for them to stick together and regulate things together. So FOXP1 sounded like an interesting gene. So Pella Duriziotis in my lab studied FOXP1 using and studied the mutation that Evan Eichler and colleagues have found using some cellular assay systems. You're going to hear a lot more about the kind of stuff that we can do in the labs with these kind of interesting assays. and other things in the next talk. But what Pella did was to take cells that she grew in the lab and then insert a normal version of FOXP1 with a special tag that means that we can visualize it in the cell. And she could see that FOXP1 is normally found in the nucleus, which is exactly what you would want. And this just shows you an overlay of this is just a blue stain for the nucleus. This is the inserted new version of FOXP1. That's a normal version that sits in the nucleus. Here's what the mutant looks like that this kid carries. So because there's a stop codon here, he's missing this fork head domain. And interestingly, there are signals at either end of the FOX domain that tell the protein to go to the nucleus. So it may not be surprising that when you express this mutated version of FOXP1, you see this staining in the cytoplasm outside of the nucleus instead of here. So that's interesting because that's a kind of functional assay we can do to test if we find a point mutation that we think is interesting, we can take it and stick it in cells. But this is where it gets a bit more interesting because this same child, remember, has had his whole genome, whole exome sequence. So we can look at other variations in the genome that may be private to this family. And there was another one that was private to the family, and it was in a gene called CatNap2, which we thought that's kind of curious because we thought we'd heard of that before. And in this case, what's strange is, or what's interesting actually, is that this kid has both these mutations. He has a mutation in CatNap2 and a mutation in FOXP1, but this CatNap2 variant is also found in the unaffected mum and in an unaffected sibling. So it's like I think Dorothy talked yesterday about kind of double hits. It's like he's taken 2 hits here in 2 genes that maybe they're in the same pathway. So what next thing that Pella did was to take some cell culture and again express the different versions of the protein and just ask what happens to CatNap2 levels in these cell systems, a bit like what Sonia had done previously for FOXP2. And this shows you sort of replication of the original FOXP2 effect, which is that if you put more extra FOXP2 or you elevate FOXP2 levels in cells, it knocks down CatNap2. Same thing happens when you express FOXP1. And this is what happened when she expressed this mutant FOXP1, which is almost like a loss of repression. You can see that it's not just back to the control, but it's actually overexpressed, if you like. I won't go into the reasons why, in the interest of time, why this might be happening, but we have some ideas about why that might be happening.
Is this part of the O'Rourke study, or is this?
Yes.
This isn't your. Okay, so it's written up.
It's part of this paper, yeah. Yes, go to this to see how we did this.
Can I ask a question here?
Yes.
Because you said this is like, these are families that have a very private mutation, and this is in families that have kids or autism and so on. Now, how do you know that actually that particular mutation is really the responsible one for this particular deficit? Because it's in essence a correlation.
Yeah, but
Unless you work out.
So the way you know, there are 2 ways you know. One is that you have to see it. You can't go on really on one variant. You have to have recurrent data. Now, it can't be recurrent in the sense that these kids don't pass on their genes, so you can't. But it's the same gene being having de novo mutations in multiple different cases of autism, and this has been, yeah. So I would argue there's two points. That's one. The other thing is this is the only de novo mutation. So under the de novo model, this is the well, maybe there's. I think this is the only coding de novo mutation that you see in these kids. So it narrows. That's how you focus in on it. Now that's assuming that that it's caused as a de novo and it's not an inherited thing or something more complicated than that. So if the de novo model is right, these kind of candidate changes are promising candidates, but you have to see recurrence in other families of different mutations. And the other thing is, I would argue, is that we need cellular assays and eventually also animal model assays as well. And this is one of the, I guess, one of the main take-home messages is it's very, all you get is correlations and we have a lot of variation to deal with, and we can't just assume that we've got a link. And actually that leads me to, well, let me just, oh yes.
How prevalent are these cases of de novo mutations? Because I know that we usually say that the
Families.
autistic siblings and were at risk.
Yeah. So I mean, I don't know what the, I don't know what the ratio is actually of these simplex kind of families versus the, because of course one of the, in the early days of studying autism, people went for, were trying to collect big families with multiple cases, and it was a lot of discussion. People were really focused on the idea of families with multiple cases, multiplex families. So, so then when this simplex study started, they have collected a large, quite a large cohort. I mean, I think now 1,000 or so, or something like that, I can't remember. But, and they're doing lots and lots of genomics on them. But what's interesting is that this doesn't tell you about these other cases where it may be, because there might also be, as Dorothy was talking about, there may also be common variations that are also putting you at risk. So this is, what this is saying is that a subset of cases of autism where it's sporadic might be explained by these kind of just odd mutations popping up. And if we can figure out what those, which of those genes are important, which are the ones that are recurrently mutated, those are the ones that we might try to understand. And one of the, I'm going to skip to this next point, because so one of the things that's interesting is how many of these genes fall into shared pathways, and I think that's going to get us a long way if we can start building pathways. And we have, in our collaboration with Evan Eichler, we went on to look at some other genes. One of the genes that we looked at was a gene called TBR1 that's expressed in layer 6 of the cortex together with FOXP2, and that has been shown to be recurrently mutated. So we took a series of, I think, 4 different mutations that were de novo mutations, and compared them to some inherited mutations, and modeled these mutations in cells, and also demonstrated that this TBR1 actually interacts with FOXP2. So again, it seems to belong to a common pathway. And this paper is coming out hopefully in the next few weeks at Nature Communications. But the bottom line here is that, again, this is another part of this pathway that we think might be interesting. And it's just a demonstration of how you should try and combine this genomics data with other things. I'm going to skip over that. So I want to end with a point on, I think I want to return to some stuff that Dorothy was talking about, genetic association studies, and how to be careful about them and how to use them responsibly. So neuroimaging genetics is this big thing that many people are excited about at the moment, and I think we should be excited about it. But at the same time, I want us to be cautious about how we approach these questions. Because, as we've already seen from the neuroimaging data that's been presented so far, these are really complicated data sets. You've got multiple, usually, even if you took a structural scan, you've got a lot of voxels in the brain that we're looking at with lots of, so you're effectively measuring lots and lots of different points, and it gets more complicated with functional data as well. Genomics, as I think I've demonstrated, can generate now these hugely complicated data sets. Even if you just have exome sequencing, you have many hundreds of thousands you might have of variants that you might be looking at. So if we have these two really incredibly complex data sets, and we try to look for associations, correlations between them, we're going to see stuff that a lot of it will be garbage, right? So how do we pull out what's sensible, and how do we design studies in a sensible way? So this is how, I mean, so these are the kind of main things that I think we should bear in mind. We should focus on defined sets of genomic variants. So we should really pinpoint candidates from other studies before we go to this level, and know the genes that, or candidate variants, and try and sort of map out their effects on brain structures. And also we should try and figure out robust structural or functional neural signatures. Really boil down your phenotype really well before you go into trying to look at the relationships. And it's not good news for exploratory analysis. It's going to be tricky for exploratory analysis, basically, because there's just so many, the space is so big. But I think if we can constrain these kind of things in a sensible way, and also have large enough sample sizes to give us enough power, then it's going to be very exciting. Now, somebody already asked me about FOXP2 common variation. So this is the FOXP2 locus with, these are the exons of FOXP2, and these lines in the middle are the introns, and it's quite a big gene itself. And I've just put 3 of the mutations on here that are known, so you can just orient yourself. So this is the KE mutation, this is a mutation in an Australian family, this is the translocation that splits the gene in this other case. So these are rare variations, just orient yourself. These black boxes are where the protein coding parts of the gene are. Over the top, what I'm showing you here are SNPs, common polymorphisms that many of you will be carrying, so G's and A's at each of these points. And you notice each of them is in introns. Again, it's a bit like this cat-nat-2 story. They don't change the protein sequence. There are no common polymorphisms of FOXP2 that change the shape of the protein or the way, well, change the shape of the protein. So we don't know, actually, what any of these variants does, or if it's functional. But people have been screening some of these variations to see whether they have effects on behavior or effects on brain structure or effects on brain function. And what's interesting here is to say, well, if we know that rare FOXP2 mutations have effects on brain structure and function, as shown by Cates and others, and we know that there are regions that are affected, like the caudate or the inferior frontal gyrus, or this is a big structure, I know, but parts of the cerebellum, we already discussed this before. Do common variants also have detectable effects? So this has been studied in a few different studies now, and this table, I don't expect you to get much from this table except to look at these sample sizes. Maybe in particular you could look at here, this is an fMRI study on 19 dyslexics and 14 controls, where they found association between this SNP in FOXP2 and quite a complicated temporoparietal inferior frontal activity on a reading task, I think. But this was a fairly complicated piece of association that they did. They got a significant association, but look at the sample size of people. Now, it's really small, is all I can say. So, and that's what we see for a lot of these, particularly ones where the brain imaging's been done, and as opposed to these are, for example, this is just a behavioral measure. Which was done in 293 patients with schizophrenia. Now, it's not surprising that the imaging studies were fairly small, used fairly small sample sizes, because it's tough to do big-scale imaging stuff. I'm nearly done. So what we thought we would do was take these SNPs and test them in relation to structure, because we don't have big enough samples yet to do functional analysis, but to test them in relation to brain structure, starting with the brain regions that have been claimed in the individual studies, and then moving to more sort of exploratory studies of more around the whole brain, but starting with these SNPs that have been claimed to be associated with particular brain regions or phenotypes, and see whether we could see any effects in a large sample. And luckily, we've sort of set up this initiative in Nijmegen, which we call Cognition Genomics or Cognomics, and these people are involved. And eventually, we're not there yet, but eventually we want to do some really large-scale stuff, integrating genomics and functional data as well as structural neuroimaging and cognitive measures. But what we can take advantage of already is, in this brain imaging genetics study, we have 1,300 brains where we have decent structural brain imaging data, and we have volumetric data on these people, and we can do VBM analysis and other things. And here you can see we tested these different SNPs, and they're all quite well represented. So we have hundreds of people in each case, and we can study these candidate SNPs. And the bottom line of all this is that we didn't see anything significant. And now we're moving to larger scale stuff, which I won't go into. So that's just a sort of note of caution for, and the nice thing is that we published it. So I'm very much in favour of doing stuff that's, I hope, adequately powered, and then publishing negative findings to back up what Dorothy said yesterday. So I'm going to finish off by saying we're in this very exciting era where we have this rapid, affordable, large-scale genotyping and next-generation sequencing, but they're data-generating tools. And so you still really need to think about study design and clever science and careful analysis, and just having the DNA sequence is not enough. Functional analyses are essential for identifying these biologically relevant variants. So sometimes we can get around this with cell-based work, but there's going to be lots of other things on the horizon. And I'm kind of paraphrasing somebody, I'll show you in a minute. At studies of complex variation, we have a great responsibility to ensure great power, which is, of course, what Spider-Man said. I may have got that wrong, but I'm not sure. I'm going to skip to, that's my special thanks slide. Lots of people were involved in lots of FOXP2 stuff, and my department, and then I will stop so that Sonia can take over.